Welcome to Jinnious Lab. Today we will cover IB Biology B1.2.1-5 proteins.
Today we're going to look at B1.2.1-5 covering protein. First thing you have to know about proteins is the basic building blocks of proteins, which is amino acids. First of all, a protein is a macromolecule composed of one or more polypeptides. The amino acids are the smallest unit, and then when they combine through bonding, they become polypeptides, which combine as well to become protein. So protein is the most complex structure, and then polypeptides, and then amino acids. There is carbon, hydrogen, oxygen, nitrogen, and sulfur in protein. And this is a very important point, which is protein structure defines its functionality. So this means if the protein structure breaks, then the protein is no longer viable and it cannot do its and it cannot function properly. We will cover this in later units, but that's what denaturation is, which is when proteins structures breaks, and this makes the protein useless. There are seven big rows of proteins, and you can memorize them by the acronym SHITSME. But often a question would only ask you to list like 5 or 6, so you don't have to memorize all of these, just memorize the ones that you are most confident that you can remember. The first one is structure, just like structural proteins, like protein in your skin and muscles that keep your structure stable, and also hormone regulation and immunity. If you didn't know, the antibodies are in your body responsible for immunity is also made of protein, and they're transport proteins and proteins that are responsible for sensation and movement and enzymatic activity, because enzymes are also a type of protein. So diving into amino acids. Amino acids are monomers of proteins that form peptide bonds to become polypeptides. Peptide bonds are just basically bonds between amino acids within the polypeptides. And there is nearly an infinite number of possible polypeptide sequences because there isn't like a limit of how long a polypeptide chain can be, and there are 20 types of amino acids. So the amino acids can bind in any type of order in unlimited lengths depending on the sequence coded by the genes. This is the amino acid structure. You will have to know how to draw this for future exams. So you'll draw the amine group on the left and the carboxyl group on the right. And there's a center carbon atom, and below there is an R group, and above there is a hydrogen atom. This is a very popular exam question, and you will be asked to draw this multiple times throughout the course, so you have to memorize this. So remember, the central carbon has 4 branches, so it has maximum bonds. And there's the amine group, NH2, and the carboxyl group, COOH. And below there's the R group. You just have to write the letter R. You don't have to draw anything else for this group. And it's important to know that the R group is what gives amino acids different characteristics because it varies by amino acids. Another diagram of a process you have to know is a condensation reaction. Condensation is creating complex molecules using smaller and simpler molecules. And the thing about condensation is that it creates H2O as a byproduct. So to remember how condensation has H2O as a byproduct, just think about when like you have a cold drink and the water, like there are droplets of water outside of the cup even though the cup itself wasn't wet before. Yeah, that's condensation. And H2O is made as a byproduct. It is a type of anabolic reaction because it synthesizes larger molecules instead of breaking down. Just as a side note, the opposite of an anabolic reaction is a catabolic reaction, which is when molecules are broken down. So just to simplify, a monomer becomes a dimer and then a polymer. So amino acids, polypeptides, and proteins. You have to know how to draw this diagram. So you just draw 2 amino acids side by side. and then group the OH and H together to indicate that H2O is made as a byproduct, and then draw the arrow, and then draw a peptide bond here. And because H2O was deleted as a byproduct, there isn't an OH or H group anymore. So yeah, this part is the trickiest part of the diagram. You have to label this as a peptide bond to get full points. So this is how the condensation reaction occurs. The carboxyl group here reacts with the amine group to create H2 as a byproduct, and then there's a peptide bond between the carboxyl and amine groups, carbon and nitrogen. And the polypeptide lengthens as more amine groups are combined with a carboxyl group, so just like there can be like an infinite chain of this. And just as a side note, polypeptide synthesis occurs in ribosomes, but you will learn about this more in future units. So this year the IB wants you to know about how amino acids are necessary in your diet. So there are two types of amino acids, essential and non-essential. Essential amino acids are not produced by the body, so you have to intake them through food. And non-essential amino acids are produced in the body, so you don't really have to consume them through food. Yeah, so it's not required in your diet. So as you can tell, it's important to consume required amino acids for proper body function. This is the list of amino acids, but you don't have to know this. This is just for reference. Again, here's a structure of an amino acid. You don't have to know all these complex structure. This is just so if you want to know more information. So the important part about this subunit is the vegan diet. As you know, it's harder to consume protein in a vegan diet because because usually plant-based protein sources have less protein than animal sources. So for example, plant-based proteins has lower leucine levels, which is a type of essential amino acid. And as a result, vegan diets require more attention to consume enough essential amino acids through sources like beans and other vegetables that are rich in protein. Moving on, as we just talked about, multiple amino acids form polypeptides, which later form proteins. The key point here is that proteins are coded by your genes. And this is possible because the genetic code is universal and degenerate. You should like sort of know this as like a phrase in your head because you will have to know this throughout like multiple future units. What we mean by universal is that the same base sequences code for the same amino acids in all organisms. If you didn't know, insulin that are used to treat type 1 diabetes are produced by E. coli bacteria, and that's possible because the genetic code that codes for insulin is the same between E. coli and humans. When we say the genetic code is degenerate, we mean that multiple codons can code for the same amino acid. A codon is a group of 3 nucleotides signaling specific amino acids, for example, like UUU and UUC. There are 64 codons, but only 20 amino acids, so there's a large overlap. So the same amino acids can be coded by multiple codons. As you can see here, UUU and UUC both code for the same amino acid. So there's a rule called the central dogma, which states that genetic codes act as instructions for protein synthesis, transcription, and translation. This will be covered much more in depth in future units, but basically transcription is when DNA becomes messenger RNA, and translation is when amino acids become polypeptide chains based on the sequence coded in the mRNA. And in school, you will probably learn how to read these codon tables and codon wheels. These usually don't have high marks, but this is very easy to read and solve questions. So yeah, it'd be very useful if you know how to read these. As I mentioned, there's an infinite possibility of polypeptide chains, and this leads to great diversity in protein structure, meaning that there is a great diversity of proteins with different functions, which is better for survival. So I talked about this in the beginning, but because protein structure is integral to its function, when the protein structure breaks, meaning if the protein gets denatured, then the protein is no longer viable. So many proteins are large molecules that are folded into a three-dimensional shape, and the shape is held in place by lots of bonds at the optimum pH and temperature. If these particular bonds are broken, then the shape of the protein is changed and then it loses its function, meaning that the protein got denatured. Like, for instance, if you get burned, your skin turns into like a weird color, or it becomes like not very good to look at because the protein in your skin that makes up your skin got denatured, and so it's like structure all got all broken down, and that's why it looks weird, and it's usually not possible to regenerate them. So denaturation is mainly caused by two factors, pH and temperature. And so that's why proteins need to be maintained in ideal conditions to perform properly. And this is also why it's dangerous when you have a very high fever for a long time, because for example, if you have like a 40°C fever, then your proteins, the proteins making up your body, which is not built to survive well in such high temperatures, will start to die, and then that can cause major permanent damage. As you can see here, like the normal protein looks like has this three-dimensional shape, but when it gets denatured, it loses all its structure and its function. The first factor is pH. So as you can see in the graph here, there's usually a very small range of optimum pH that the protein can function in. The hydrogen ion in acid reacts with the R group bonds, so it breaks the intermolecular bonds in the R group, which breaks the amino acid structure, and thus it denatures the protein. So for example, pepsin, which is a stomach enzyme in your stomach, which is active in the pH of 2, won't be active in like neutral pH in the rest of your intestines because it will get denatured in alkaline conditions. The second factor in denaturation is temperature. As you already probably know, heat increases the vibration of molecules, and so if the heat is too high, then the molecules vibrate so hard that the bonds between the amino acids break and the protein structure starts to break as well. This is almost always irreversible. And you can see here, when the temperature becomes too high, the stability of the protein structure drops suddenly and the protein gets denatured. For example, you would have noticed that egg whites are actually translucent before cooking, but after you put heat on them, they turn white. And that's because the protein called albumin in egg whites denature when coming in contact with high heat.
For IB45 Jinnious Lab, don't forget to subscribe and like.